Okay, lecture 11, the topic of our discussion is southern blot hybridization. So again, it may be a little outdated technology or technique, but the principle is pretty important, so we'll talk about it. And at the end, we'll talk about the modernized form of this technique. We use a process called gene chip assay, which is developed based on the same technology. So still, it is being used when we screen gene expression pattern using multiple probes. So you need to understand the principle of this southern blot hybridization first, because this is a pretty important technique. Okay, so when you say hybridization, especially using a probe, what does that mean? So when you say you are hybridizing a DNA, piece of DNA against some other matching DNA, what that means is you are trying to create a binding between a short stretch of your probe. A probe is just like PCR probe, a single-stranded DNA that has a unique sequence, right? You are trying to find a matching DNA, or RNA could be the target as well, by forming the hydrogen bonds between the bases. Okay, so if this is considered as a probe, when it binds with another strand that has the complementary matching sequence, yeah, that is a hybridization. Okay, so then why is it not considered as the renaturation? Because there is a difference in their sizes, right? So this probe is much shorter than the matching DNA strand. So this is not a renaturation because they have a difference in their sizes. Under this condition, it's the same way. So this probe is binding with this DNA, but here it's not a perfect match. There's a one nucleotide mismatch there, but still it's a hybridization. Okay, same goes here. The probe is binding with this with a less than perfect match, and same thing here. So these are all considered as a hybridization process. Okay, hybridization process. So when you have a probe, which is a short stretch of single-strand DNA, when it binds with a DNA strand that has a matching sequence, we consider that a hybridization. Okay. Okay, then the question is, why is that important? Where is that used for, hybridization process? So hybridization process is important because through that process we can understand the similarity between two nucleic acid molecules. When I say similarity, what does that mean? It could mean the sizes, or it mostly means the similarity in the base pair sequences. Okay, so if one stretch of DNA is paired up with another stretch, okay, there should be enough complementary matches to be able to pair up, right? So when a probe or primer is binding with its target, yes, there should be enough complementary match. Okay, when it binds together, making that hybrid, then yeah, hybridization is formed based on their sequence match. If you can form that hybridization, then that means there is enough sequence similarity. So that's a demonstration of complementary sequence match. Okay, another place where it's used for, let's say you're trying to look for a homologous gene. Homologous gene, what does that mean? You know, between the species barriers, let's say you have a gene that's sequenced from a human species. Okay, that gene is serving an important function. You want to study that if that gene is existing in other species, maybe chimpanzee, or maybe a mouse, or maybe distantly related, like even like plants, or different animals, or different bacterial prokaryotic cells. You want to see if that gene is located in different species their genome. Okay, so that's what I mean by homologous genes. How do you do that? You can develop a probe that has a unique sequence of your gene of interest. Okay, it doesn't have to be your entire gene, maybe a small segment of your gene, and then you use that as your probe and try to hybridize against other animal, plant, or bacterial cells genome and see if it binds, if it forms a hybridization. If it does, then most likely that hybridization is formed between your probe and the target segment of a genome. Most likely will have the homologous gene in them. Okay, so it's used for hunting of homologous gene. Okay, using a probe. Gene cloning, of course, just like PCR, this hybridization process can be used to not only identify homologous gene. And once you identify it, what do? You understand where the gene is located and probably cut and paste. That's a gene cloning, right? Create a new recombinant construction and move it to a different location. You can do that, okay? And then medical diagnosis, okay? So, when you do maybe COVID-19 screening, okay, what is being done? So when they take the swab sample from deep in your throat, they can use the hybridization process using maybe a PCR probe and see those PCR probe can bind and hopefully amplify out the virus RNA or DNA, the replicate DNA or viral RNA. So it is used in the medical diagnosis. Okay, so hybridization process is important. Okay, so the original technique is named after its inventor again. So Alfred Sutton, E.M. Sutton was the name of the inventor. It's done quite a while ago, 1975. It's a long time ago, almost like a half century ago. The scientist developed this method. Okay, so originally it was used for mapping of the genome. Okay, and then identifying location of those genes using a probe. That was how this method was developed. Okay, so the process-wise, first at that time in 1970s, okay, since the PCR technique was not readily available, so they had to deal with something called a genetic library. So I hope you are familiar with that term, genetic library. What it is explained in this diagram. So if you have a genome of certain species, could be human genome, it could be a bacterial genome of any kind that you are trying to study. Okay, that genome is a huge DNA, chromosomal DNA, right? That can be cut up into smaller pieces by using an enzyme, precision enzyme, that can cut maybe every 4 base pair, not every 4 base pair, maybe it recognizes specific 4 base pair sequence. Okay, and if you use that enzyme, you can generate lots and lots of code. So maybe you can generate about thousands of smaller fragments of the DNA from the genome. Once you have those fragments generated, and those individual fragments can be recombined with a plasmid DNA vector. So individually, all 1,000 fragments will be recombined with this plasmid vector. So you can create thousands of recombinant DNA from the genome that's cut into thousands of pieces. And the plasmid DNA copy, you may have thousands of those copies, and they are all ligated together. So you're creating about thousands of different recombinant DNA constructs. And those recombinant DNA constructs can be moved into a living cell, like E. coli. So E. coli cell will take up maybe one recombinant DNA at a time. So you can create thousands of E. coli DNA with different recombinant DNA constructs. Okay, and you can collectively have all those cells grow up in a medium, and then you can maybe even keep them in a frozen vial. So that is the genomic DNA library. Basically, your genomic DNA is cut into smaller pieces and is sorted out in this plasmid DNA vector, and then now they moved into the storage place, which is the E. coli cell. Okay, so E. coli cells can be frozen, and they can be stored in -80-degree freezer, and then whenever you need the genomic DNA of that species, instead of going back to the species, try to get this DNA isolated, you can go back to these E. coli cells, okay, that are carrying these recombinant DNA. You can grow them up and wake them up from the frozen condition by moving into the liquid medium, growth medium, and grow them. Then you can replicate thousands of more cells, and from those cells you can make more of this DNA. Okay, so you can isolate this DNA and do whatever you want to do, carry out your research using these as your DNA resources. So it will continuously provide you with the source of DNA that you will need to study on. Okay, so that is the genomic DNA. Okay. So once you have the genomic DNA, okay, out of these thousands of cells that may carry different types of recombinant DNA, okay, you may wonder, so let's say you are interested in maybe a certain gene within the genome, okay, and your question is, okay, that gene fragment may be cut off from this genomic DNA piece, and then it might have been ligated with this plasmid DNA, and then it might have moved into one of these thousands of cells, right? Then the question is, how can you identify a E. coli cell that has the right recombinant construct with the piece of DNA that has the gene that you're looking for? How can you identify that? Okay, that's where this southern blot hybridization process will come in, and it'll be able to help you by identifying the piece of DNA that's located within one of these thousands of cells. Okay, so we'll talk about the process. So let's assume that this is a probe that has the gene sequence. Okay, and if you can somehow have that probe hybridize against the DNA fragment that has the matching sequence, because that fragment has the gene that you're looking for, okay, then that hybridization process will allow you to identify the cell that carries the correct recombinant DNA structure out of these thousands, right? Okay, that's the theory. Okay, then in order for you to do that, we saw this diagram. It says yes, that's a probe, but that probe has this star on it. So what does that star mean? The star means, yeah, probe is labeled with something, okay, some chemical or something that will give you a signal. Okay, so when you do this hybridization, you cannot visualize this process otherwise, unless you have some kind of signal that's being generated by this probe. Okay, to detect this hybridization process, you have to link your probe DNA with something that will give you a strong signal. Okay, that something could be maybe the radioactive isotope. For example, the phosphate isotope can be attached to your probe DNA. Okay, let's say this is your probe DNA. Okay, and then maybe ATP that's labeled with radioactive isotope phosphate. That phosphate can be transferred from this nucleotide to your DNA. Okay, through an enzymatic reaction called kinase activity. So it's a phosphorylation. So phosphate is transferred from this ATP to the end of your DNA. Okay, so the kinase enzyme can do that. Then once that's done, then your DNA is labeled with radioactive phosphate. So it's going to give you a strong radiation signal. Okay, so that's one way of labeling your probe. Once you have this probe, then you can use this probe, radio-labeled probe, for your process. Okay, so that's one way of doing that. Another way of doing that is maybe you can cut your probe with an enzyme just like EQAL1. Okay, then you will have these crisp ends generated at the end of the fragment, right? And to that fragment you can do the fill-in again, because by adding the DNA polymerase and the nucleotide, you can refill that cut up end. When you do that, you provide radio-labeled nucleotide. Let's say the A is radio-labeled, then the enzyme polymerase will grab those A and insert them into them. So now once that's done, this DNA is now radio-labeled as well. So there are a couple of different ways of radio-labeling your probe. Okay, whichever method you use, yes, the answer is you can radio-label your probe for your analysis. Okay.